According to YouGov.com, 7 out of 10 people say they would miss vision the most over the other 4 senses if they lost it. Eyes are amongst some of the most energetically expensive organs to develop, and 70% of all sensory receptors are in the eyes. That's the majority, within the 2 little balls in the head compared to the whole body. 50% of the cerebral cortex is involved in visual processing. That's so much. Since we clearly value our eyes so much, and there's so much devoted to them, why not learn a little about our eyes? A few commonly confused parts of the eye are the retina, optic disc, and fovea. The retina is the layer of tissue that contains all sensory receptors for vision, rods and cones. These rods and cones are the photoreceptors, responsible for receiving photons, light particles, and initiating the process of phototransduction, the process of converting light signals into their action potentials. The retina is the entire layer. The RE at the beginning of retina will help you remember that it's the retina that's the layer on the eyeball, not necessarily a certain structure. The next 2 mentioned will be structures. The optic disc is where the axons of the neurons coming from the retina converge to form the optic nerve, which will then head towards the brain. If you navigate about 3.5 millimeters above the optic disc, you'll find the fovea, which has the highest concentration of cones and therefore greatest visual acuity. We need our fovea for reading, driving, watching YouTube videos. We want to keep our fovea forever. Now, let's go over some important structures of the eye anatomy. The choroid layer, which lies just superficially from the retina, handles supplying blood and nutrients to the retina. The sclera is the outermost layer of the eyeball. It's white in color and gives the eye its overall shape and form. The iris consists of 2 layers of smooth muscle that encircles the pupil, thus allowing the pupil to dilate and constrict in size. The purpose of the iris changing the pupil size is that it controls how much light is allowed into the eye. The importance of that we'll discuss later. The iris also determines your eye color, be it brown, blue, green, or hazel. Higher melanin levels in the iris equate to a darker color, such that brown eyes have the most melanin and blue eyes have the least melanin. As you can see, learning anatomy is pretty straightforward. Let's get groovy. The cornea and lens work in tandem to focus light onto the fovea of the retina, ensuring best vision possible. Let's begin on how the cornea and lens focus light. When light passes from one medium to another, it is refracted or bent to some extent. The higher the refractive index, represented by N, of the material light is passing through, the more the light rays will bend towards the normal line, seen here. Applying this concept to the eye, we see the cornea and lens are responsible for bending light so that the image in question is cast directly on the back of the retina at the fovea. Since the cornea is more dense than air, light passing from air to the cornea is naturally refracted to a moderately high degree because it has a moderately high refractive index towards the fovea. In fact, the cornea does the majority of the refraction of the eye. However, the lens is adjustable and can change shape to provide greater refractive power, a process called lens accommodation. The changing of lens shape occurs by contracting or relaxing the ciliary muscles and adjusting the tenseness of attached zonular fibers. For example, the eye viewing a distant object and a near object would both be seeing clear images of that object. However, the eye witnessing the distant object would be receiving nearly parallel light rays passing through the cornea. As such, the lens only weakly accommodates, becoming more convex or spherical, to refract the light rays onto the fovea. In other words, the lens doesn't have to work as hard as it would when focusing on a closer object, which, as we can see in the eyes viewing the near object, experience more diverting rays. For near vision, The lens has to accommodate and become more and more convex as the object is moving closer. The lens and cilia muscles get fatigued easily, so it's important to follow the 20-20 rule. That is, take a break every 20 seconds, look at something 20 feet away every 20 minutes. The lens can also accommodate so much before an object becomes blurry. This is how we measure visual acuity with the Snell and eye chart. The basis is this. XY vision means the person can see something X feet away while the average person can see that Y feet away. So for 20/60 vision, this means the person can see something that is 20 feet away while the average person can see it 60 feet away. Another factor that we must consider when focusing on an object is the amount of light available that is allowed to enter the eye and hit the lens. This is controlled by the pupillary light reflex. The pupillary light reflex is controlled by 2 rings of smooth muscle. Pupil constriction is controlled by the parasympathetic system, intervening circular muscles whose fibers run around the circumference of the iris. The sympathetic nervous system controls the radial muscle fibers, which expand the opening of the pupil, which is dilation. Pupil constriction decreases the amount of light entering the eye, thus ensuring that photoreceptors don't get overwhelmed with light and preventing the complete whitewashing of vision. Seen here. Pupil dilation increases the amount of light entering the eye, thus ensuring that photoreceptors get the right amount of light and allowing for the best vision possible and preventing vision from being too dark. Seen in the left image. Another note, ever wonder why people who gamble typically wear sunglasses while gambling? It's because they took human physiology and they know that when they draw that high-level playing card, their eyes will reflexively dilate as the sympathetic nervous system is stimulated, giving themselves away to other players. Our eyes aren't perfectly spherical, but when there is a noticeable squish, that condition is called myopia or hyperopia. You can remember this by looking at the length of the word. Myopia is a much shorter word. The first and last letters are nearer, which correlates to nearsightedness, which means you can see near, but not far. That's how I remember it. Hyperopia is a longer word. The first and last letters are farther, thus farsightedness. You can see far, but not near. Stigmatism occurs when both eyes have different degrees of myopia or hyperopia, as seen in approximately a third of adults, although technically we all have some degree of astigmatism. After all, our eyes can't be exactly identical. Let's switch gears to learning how phototransduction cascades work. Strangely enough, light must travel through the bundles of neurons to reach the pigmented retinal layer where light is absorbed and triggers photoreceptors. The signaling pathway that transmits back towards where the light came from, passing the bipolar cells and then to the ganglion cells. It's the ganglion cells that transmit the action potential to the optic nerve. Looking at the rods, you would expect that when the rods are depolarized, the action potentials will travel down the axon towards the bipolar cell. However, that is not entirely true. It is indeed an unusual case for our sensory system. The rods are unique in that in dark, they are depolarized. In light, they are hyperpolarized. So the rods are essentially activated or turned on in dark circumstances. They are inhibited or turned off by light. Let's start with a broad overview. In the dark, when the rod is depolarized, more positive inside, voltage-gated calcium channels open and calcium ions are moving into the cell, causing the exocytosis of neurotransmitter, inhibitory glutamate. Inhibitory glutamate binds to the receptor on the bipolar dendrites, causing hyperpolarization, inside is more negative, and therefore inactivation. As a result, the ganglion cell will not become activated. everything is completely dark and there's no vision. In the light, the rod becomes hyperpolarized, so more negative inside, which decreases the amount of inhibitory glutamate that gets released into the synaptic cleft between the rod and bipolar cell. Less inhibitory glutamate allows the bipolar cell to depolarize, more positive inside, and generate an action potential which ultimately release excitatory glutamate. Excitatory glutamate binds to the receptors on the ganglion cell dendrites, causing depolarization and therefore excitation. Action potentials are generated and they travel towards the optic nerve and onwards towards the brain. There is light and vision occurs. Now, let's zoom in closer on how the cell physiology of rods and vision works. The rods are about 20 times more abundant than cones. 120 million rods inside an eye versus 6 million cones inside of an eye. And they're unique in that they contain shelf-like discs of photopigment, rhodopsin. Cones, while less abundant, are more important for high acuity vision and are more important for human vision. What we discuss in this video revolves around rods. Very similar process occurs with cones, just with more narrow ranges of wavelengths of light. When light hits rhodopsin, transducin detaches, becomes active, and activates cGMP phosphodiesterase, which breaks down cGMP abundant in the cytoplasm of the rod. The subsequent decrease in cGMP concentration within the cell causes the cGMP-gated sodium channels to close because their activating ligand, cGMP, is greatly lowered. The result is hyperpolarization of the rod, inactivation of the rod, and decreased release of inhibitory glutamate. As such, the bipolar cell is not overly inhibited, and action potentials are generated, and excitatory glutamate is released. Ganglion cells become excited, generate their own action potentials, and signals are transmitted along the optic nerve toward the brain. Vision ensues. When rods are in darkness, rhodopsin cannot activate transducin, which cannot break down cGMP. The buildup of cGMP in the cytoplasm of the rods mean the cGMP-gated sodium channels are open, allowing a rapid influx of sodium into the cell, thus depolarizing them. This depolarization allows voltage-gated calcium channels to open, and thus a high level of exocytosis of inhibitory glutamate. The high level of inhibitory glutamate will strongly inhibit the bipolar cell and prevent any further visual signaling. Now let's finish with a few final notes on vision. We humans, like many other animals, use contralateral and ipsilateral visual pathways. So neurons of optic nerves from both eyes follow two pathways heading to the thalamus and primary cortices. Ipsilaterally, neurons transmitting visual signals from the right eye to the right primary visual cortex, while visuals from the left eye travel to the left primary visual cortex. At the same time, contralaterally, visual signals from the right eye travel to the left primary visual cortex, and visual signals from the left eye travel to the right primary visual cortex. The crossing over occurs at the optic chiasma. The contralaterally and ipsilateral visual pathways contribute to our and other animals' advanced depth perception. So animals like horses, rabbits, fish don't have this depth perception because they lack the dual pathways for visual signals.